Do you know that there's this creature that lives inside our house that is minuscule in size but is actually pretty dangerous, and researchers suggest that it can harbor to at least 65 illnesses that infect people? So if that's not enough to creep y'all out, let's try to look into some interesting but weird facts about this very common but engrossing creature. Okay, so first fact is that they defecate a lot. So their digestive system can move quite quickly. So it is speculated that they defecate every time they land, even on their next meal. Second, when they land on a tasty meal, it is all down to their taste receptors being located on their lower legs and feet. Third, they have compound eyes. So their intricate eyes provide them with nearly a 360-degree field of view, which allows them to see behind themselves. And lastly, they are able to process what they see and react accordingly at amazing speeds. To put things into perspective, our brains process around 60 images a second, whereas to them they can process around 250 in a single second. Okay, so the main question is, what are these potentially killer creatures that live among us? Okay, so to not waste any more of our time, let us now introduce this infamous creature, or as a matter of fact, an insect known as the Muxa domestica, or the housefly. Okay, so the classification of a housefly, so their kingdom Animalia, phylum Arthropoda, class Insecta, order Diptera, family Musidae, genus Musca. So it is a fly of the suborder Siderhapa. It is believed to have evolved in the Cenozoic era, possibly in the Middle East, and has spread all over the world as a commensal of humans. Okay, so for morphological characteristics, they are 6 to 7 millimeters in size, they are covered with short hairs, they are strongly convex in front and flat, and slightly conical behind. They have 3 simple eyes and a pair of short antennae. So the females, they tend to be larger winged than males, while males have relatively longer legs. Compound eyes almost touch in the male, but in the female, they are more widely separated. They have retractable, flexible proboscis with an enlarged fleshy tip, the labellum. So their thorax, it is a shade of gray, sometimes even black, with 4 dark longitudinal bands of even width on the dorsal surface. So their abdomen is gray or yellowish with a dark stripe and irregular dark markings at the side, and they have only 1 pair of wings. Okay, so the housefly life cycle closely mirrors the most insects. So a basic cycle that begins with an egg, then develops to a larvae phase, a pupa phase, and finally into an adult. So during a warm summer, optimal conditions for a housefly. Once a male housefly chases down and fertilizes a female counterpart, she's ready to lay her eggs. Houseflies are solitary creatures. Like the rest of the insect world, males and females do not stick together after mating, and unlike nesting insects, females do not care for or protect their eggs. Females simply leave the eggs where they will be safe from predators and have plenty to eat upon hatching. Okay, so a female housefly is capable of laying up to 150 eggs in a batch. Housefly eggs resemble individual grains of rice, and within a day, housefly eggs hatch into larvae, known as a maggot. So maggots are legless white insects that feed from the egg-laying site for 3 to 5 days, and during this time, maggots molt. So fly pupae are similar in function to butterfly cocoons. Their hard brown shells protect the inactive developing flies. So over the course of 3 to 6 days, the pupae develops legs and wings and ultimately emerge as full-grown houseflies. So within 2 to 3 days, the female houseflies are capable of reproduction. Okay, so medical importance. So flies commonly develop in large numbers in poultry manure under cage hens and in situations where plumbing is lacking, such as open latrines. Serious health problems can develop, especially if there are outdoor food markets, hospitals, or slaughterhouses nearby. Pathogenic organisms are picked up by flies from garbage, sewage, and other sources of filth and then transferred on their mouthparts through their vomits, feces, and contaminated external body parts to human and animal food. Okay, so although this fly does not bite, The most important damage related with this insect is the annoyance and the indirect damage produced by the potential transmission of pathogens associated with this fly. Of particular concern is the movement of flies from an animal or human feces to food that will be eaten uncooked by humans. Also, when consumed by flies, some pathogens can be harbored in the mouthparts or alimentary canal for several days and then be transferred when flies defecate or regurgitate. Okay, so among the pathogens commonly transmitted by houseflies we have Salmonella, Shigella, Camelobacter, Escherichia, Chlamydia, and many other species that causes illness. So these flies are most commonly linked to outbreaks of diarrhea and Shigella. But they are also implicated in transmission of food poisoning, typhoid fever, tuberculosis, and parasitic worms. Okay, so now let's move on to control. Okay, so first up, we have reduction or elimination of fly breeding sites. So solid concrete floors with drains should be constructed, dung should be cleaned out, and floor should be flushed daily. Leaking water pipes should be repaired, and dung should be removed, and the floor should be flushed at frequent intervals. Okay, so next we have reduction of sources that attract flies from other flies. So flies are attracted by the odor emanating from breeding sites. In addition, they are attracted by products such as fish meal and bone meal, as well as sweet-smelling fruits, especially mangoes. So attraction to waste can be prevented by cleanliness, the removal of waste, and its storage under cover. Okay, so next up, we have prevention of contact between flies and disease-causing germs. So the sources of germs include human and animal excrement. So measures to eliminate fly breeding also reduce contact between flies and germs. The most important are the installation and the use of proper latrines and toilets, the prevention of contact between flies and sick people, and the prevention of access of flies to slaughter offal and dead animals. And lastly, we have protection of food, eating utensils, and people from contact with flies. So food and utensils can be placed in fly-proof containers, cupboards, wrapping materials, etc. So nets and screens can be used on windows and other openings. Nets can be placed over babies to protect them from flies, mosquitoes, and other insects. So flies that enter screen rooms can be killed with traps, sticky tapes, or spade sprays delivered from an aerosol spray can. Okay, so now that we know about houseflies, let us now meet another insect within the same Mucidae family. And like before, let me state some interesting facts about the species before its proper introduction. Okay, so first fact is that they're often mistaken as houseflies. The resemblance can be quite confusing. Second, it is a cosmopolitan insect, meaning that it can be found worldwide as long as suitable food and water conditions can be met. Third, they love barn animals, as it is almost anywhere that horses, cattle, and other agricultural animals can be found. And lastly, they are strong flyers, and dispersion from one livestock facility to the next is common. Okay, so without further ado, this mystery insect is what we call the Stomoxys calcitrans, or commonly called as the stable fly. Okay, so the classification of a stable fly comprises of its kingdom Animalia, phylum Arthropoda, class Insecta, order Diptera, family Mucidae, and genus Stomoxys. Okay, so for morphological characteristics, the stable fly adults are similar to the housefly in size and coloration. The length of an adult stable fly is typically 5 to 7 millimeters in size, and they can be identified by 4 characteristic longitudinal stripes across the thorax as well as several dark spots on the top of abdomen. On the vertex and in front, there are 3 ocelli and 2 large compound eyes. Sexual dimorphism occurs in this species, and there is more distance between the compound eyes in females. The proboscis of the stable fly is black, long, and thin, protruding from the front of the head. The palps are 1/3 of the length of the proboscis, and stable flies have long, bayonet-like mouthparts for piercing skin and feeding on the blood. Okay, so stable flies breed in moist, decaying organic matter. The adult female lives for around 7 to 10 days in the field, and during this time, she lays multiple clutches of eggs. So egg clutch may contain 60 to 130 eggs, which are laid in small groups within a subtle substrate. So each female fly may lay up to 800 eggs in her lifetime, with each clutch requiring a separate blood meal. So eggs hatch in 12 to 14 hours into first instar larvae, which feed and mature to 3 instars in 12 to 13 days at the optimum breeding site temperature of 27 degrees Celsius. So third instar larvae transform to pupae within the puparia. So the adults develop inside and then emerge from the puparia. The average stable life cycle in the field ranges from 12 to 20 days, depending on the environmental conditions, but is usually around 28 days. Adults female can start lay eggs 5 to 8 days post-emergence. Okay, so now let's move on to medical importance. So they affect humans in two ways, livestock production and disease. So the accumulation of stable fly leads to a certain degree of anemia, weight loss in cattle, and reduced milk production in dairy cattle. So in this way, escalcitrants cost the US millions of dollars. Okay, so stable fly bites are extremely painful and the flies are very persistent. They often ignore swatting, stomping, and other tactics used by animals trying to avoid bites. However, unlike many other blood-feeding insect bites, on humans the bite site does not appear to get irritated and bites rarely results in allergic reactions. Okay, so for ox, horses, and sheep, escalcitrans is a vector of Trypanosoma casalva. So the parasite causes the disease known as suma. For domestic animals and humans, it is a vector of anthrax. So the disease can cause a number of different symptoms, including lesions in the lungs and brain. So it is also a vector of the following. So we have swine erypsiles, equine swamp fever, African horse sickness, and fowlpox. Okay, so for control, so the stable flies prefer to feed outside and are rarely found feeding or resting indoors. Stable flies observed on their animal host are usually feeding, but they leave the host immediately after feeding is completed. Consequently, infestation in the early stages may not be noticed until the situation is well above the threshold where economic damage is inevitable. That is why monitoring is important for determining when management is necessary to prevent an outbreak situation. So the preferred habitat of stable flies is herring feeding sites, the reservoirs of feeding hay in fields. However, breeding may occur in any type of moist, decaying organic matter such as silage, crop, hay, grain, manure, and many others. Okay, so to reduce fly breeding, spread crop residues and animal manures thinly so they dry quickly. Clean up spilled food and rotate hay feeding areas in fields. Reducing the composition of organic matter around animal facilities also will reduce the presence of other localized fly problems for the health and safety of the animals, the neighbors, and the local community. Okay, so alternative control measures include the use of traps, insecticides, and insecticide treatment of stable fly resting sites or cattle. Insect repellents may also be used to provide some relief to humans and other animals. For example, natural repellents containing citronella are often used on horses.
Now we're going to talk about the Latrine-Flies. Well, knowing further its classification, its genus is Phania and belong to the family Phanidae, but some would place it in the family Muscidae. Its kingdom is Animalia, phylum is Arthropoda, class is Insecta, order is Diptera, and well, you know the rest. Now that we have established its classification, we're going to talk about more of how the flies of the genus Phania resemble somehow to houseflies. They're a little bit smaller, sizing about 5 to 7 millimeter. And while we're on the subject on differentiating them from the houseflies, latrine flies are distinguished from houseflies by having vein 4 on of the wing that is almost parallel to vein 3. Whereas in Musca, it bends upwards and almost touches vein 3 at the wing apex. And and in Muscina, the vein 4 also bends upwards, but not to such an extent. The arista arises from the third antennal segment and would lack hair completely. On the other hand, in both Musca and Musina, there are hairs on both upper and lower sides. Moving on to getting to know more about the latrine flies, there are two common species of Phania that are known to be a minor medical importance. First is the little housefly or the Phania canicularis, which occurs worldwide and well it's commonly encountered in houses, but the latrine flies or the Phania sclaris has a holarctic distribution and is uncommon in houses. Adults of the little housefly have three rather faint dark longitudinal stripes on the thorax, whereas the latrine flies have two such stripes. Well, it keeps me on deep thought on how this type of fly develop. And well, it's a good thing that we are now going to talk about its life cycle. The larval development takes about 7 to 12 days but may be extended if habitats start to dry out. The puparium is brown and is similar in shape to the larva. After 7 to 10 days, the adult fly emerges. The life cycle lasts about 1 month, but under some conditions. It can be completed within 13 to 22 days. Their eggs hatch after 1 to 2 days. Fania larvae are unlikely to be confused with the larvae of other medically important flies. The latrine fly usually lays her eggs in feces, and the larvae of Fania species prefer wetter breeding places. And when this little house fly becomes an adult, they often enter houses and tend to hover around the head in an annoying manner, unlike the house flies. They do not settle on people or on their food. Many of the pathogens transmitted by the houseflies probably spread by the Phanes species, and in cases of oral and urogenital myiasis, their larvae are sometimes found in stools, but you have to take note that true intestinal myiasis does not occur in humans. It can be deposited on food, but also on urine-soaked bedding of humans and animals, such as decaying piles of grass, human and animal body waste, and well, it can be seen in poultry litter. Well, it's a good thing that the same control methods apply to species of Fania as to Musca, but keep in mind that particular attention should be given to the prevention and removal of Fania species and breeding in latrines.
So now let's move on to our gap. So the question was suggest the best control method and provide journals to prove them. So I have gathered two journals that can answer this question. Okay, so the first journal is from Maury Kircher with an entitled journal called Best Management Practices for Fly Control. And these are her recommendations. Okay, so since stable flies require a combination of organic matter, animal waste, and moisture to reproduce, they can reproduce in about 24 days. So the best management practice to control this fly is sanitation. In other words, if you can break the cycle of egg production, you can make rapid progress in controlling fly numbers. So the best way to do this is by removing spilled feed, cleaning barns every 7 to 10 days, and minimizing water spillage. Another method is the use of dust bags, baits, and sticky tapes. Another newer method is using parasitic wasps. So this small insect will attack fly larvae and prevent larvae from hatching. So this is an effective tool provided you use the right parasitoid and you do not wait until the stable fly population is out of control. Okay, so next up we have houseflies. So control measures for this fly are very similar to the stable fly. Most of these flies depend on organic matter to reproduce, and breaking the cycle is accomplished by using sanitation methods and keeping barns as clean as possible. Minimizing water and feed spillage can be very helpful in controlling this insect. To note, when controlling flies, that means cleaning corners and areas that you might not reach with skid loaders or power equipment. Once again, a parasitic wasp is an option along with larvicides, sticky tapes, and fly baits. So for my second gap, we have biological control of houseflies and stable flies by means of inundative releases of Sphalangia cameroni. Okay, so among the natural enemies, pupal parasitoids have been studied intensively as potential control agents of houseflies and stable flies in livestock installations. In the last 5 years, control of houseflies and stable flies by means of pupal parasitoids has been given high priority in Denmark, guided both by a political goal to reduce the use of insecticides and by the growing problem of resistance to many of the commercially available products. Previous surveys in Denmark have shown that Sphalangia cameroni perkin is the most active indoor parasitoid of nuisance fly pupae, and several studies have demonstrated that this species is the one that surges most deeply in the larval substrate and appears best adapted to the dim light conditions prevailing in livestock units. Okay, so 3 farms located 40 to 50 kilometers north of Copenhagen were included in this study: a dairy cattle farm A, pig farm B, and another pig farm C. So Sphalangia cameroni was released by filling the contents of a rearing bucket into small mesh cages, which allowed the adult parasitoids to escape, but retained the puparia from which a few more parasitoids were expected to emerge. So the abundance of houseflies and stable flies generally increased from June through to September, followed by a slow decline towards December. So this pattern was seen in the control years on all 3 farms, despite the occasional use of pyrethroids on farm A and B during the peak period, which partly blurred the general picture on these farms. However, weekly releases of S. cameroni from April to September and October resulted in significant suppressions of housefly populations to below nuisance level, whereas no effect on stable flies was found. So parasitism was significantly higher in the released years compared to the control years, but was below 25% average over the fly season for each farm. So the study indicated that biological control of houseflies can be an efficient alternative to chemical control.